Bom dia, boa tarde, boa noite aos internautas que nos acompanham neste programa Nanotecnologia do Avesso. Hoje no seu número 478. Fazemos este programa dedicado ao público não especialista, porque é este público que paga as pesquisas sobre nanotecnologia realizadas no Brasil. Também fazemos este programa porque é aqui que nós podemos discutir os impactos da nanotecnologia na sociedade, no meio ambiente, na economia, na saúde, no direito, etc. Coisas que não são feitas aonde se faz nanotecnologia no Brasil. Por fim, fazemos este programa porque acreditamos que ser livre é ser bem informado e a divulgação científica contribui, colabora para o aprofundamento da democracia e da cidadania no Brasil. Hoje, conforme tínhamos anunciado, estamos conectados com o professor Kenneth Shea, diretamente da Universidade de Califórnia, IRVINE, e que tem trabalhado com uma coisa bastante importante para a gente, que é a relação da nanotecnologia, possivelmente aplicada à picadura de cobra, não é? Então esse é um tema que nós não tratamos ainda em nossos programas, não tem a oportunidade de tratar hoje. Eu vou pedir ao professor Ken que se apresente e depois ele vai iniciar a apresentação de hoje. Ok, Professor Ken, could you please introduce yourself? Yes. You.
give some indication of the scale of things, just what is a nano. Since we'll be talking about nanomedicine, we'll introduce the concept of a magic bullet.
Okay.
And that will allow us...
Professor, you have a screen for this or not? Because
Pardon me?
You have a screen for this that you are talking for us or not? Because there is not in the screen.
It is not on the screen?
No.
Oh, okay, so hold on.
Yes, about the share.
Yes, okay, so let me... I do want to share the screen, and
yes, now is no.
Now you can see the screen.
No, the screen is not, because there is yes. Now yes, yes.
Now yes.
Yes.
Okay.
Okay.
And so. Please, if for whatever reason the screen disappears from my slides, please interrupt me, and I'll try to adjust and correct. Okay?
I will do it.
Very good. Okay, so we'll talk about the magic bullet, and this will provide us with an opportunity to introduce antibodies and have some discussion about antibodies. That brings us to considerations of the current pharmaceutical drug market, in which antibodies play a very important role. We'll talk about emerging areas of pharmaceutical science, that is the biologicals, and that's what I'm referring to when I talk about antibodies. And then we'll talk about our research, which is an exploration of developing alternatives to traditional biologicals or antibodies, so-called plastic antibodies. Plastic is a word that we use because the materials, the nanoparticles that we make, are synthetic organic polymers, just as plastics. They have quite different properties, of course. And then finally, I'll talk about our efforts to develop a broad spectrum antidote for snakebite. Has the screen changed?
Yeah, no, you can continue.
Okay, fine. So I've changed slides now, and we introduced this concept of nanomedicine. What is nanomedicine? And nanomedicine is the medical application of nanotechnology. So now we have to ask the question, what's a nano? And my intent here is to give you some idea of the size of the materials that we're working with. I know this entire lecture series put together by Dr. Martin has focused on this, but I always like to introduce this concept to a general audience at a very fundamental level. And so if we ask the question, what is a nano? We see here a ruler with length scales on it. And it actually encompasses a very small part of our universe in terms of the size. Because at the very top, we actually have cells. These are red blood cells. And typically, these are 2 to 5 microns. A little bit larger than that is a millimeter. And I want to call attention to the fact that a millimeter, which is a quite small unit of length, is 1 million nanometers. So we're really going to be talking about small things. As we come down to smaller and smaller objects, we might focus on virus particles. And those typically within the domain of 10 to 300 nanometers. Keep in mind again, a nanometer is 10 angstroms. Now, I'm a chemist, and so my comfort zone is in the molecular and atomic level. And so we've always been, and chemistry has always been, in the nano regime. But there have been developments over the last 25 years or so that has called out an emphasis on this concept of nano. Coming down a little bit further, typical molecules like this carbon buckyball is about 1 nanometer across. And so molecules, such as this DNA, they can have a rather long length, but their width of a DNA duplex in this particular case is approximately 2 nanometers. So this gives some indication of where we are with regards to nano. Now, I had a former student who works for a company that develops the next generation tools that make our processors in our computers and laptops. And he took a series of photographs and enlarged them to give us some concept, again, of what is a nanometer-sized feature. And so this process starts with a photograph of a little tiny sugar ant on his finger. And once again, this is an effort to call attention to the fact of just how small is a 100 nanometer feature, which, as you'll see later in the talk, is where we're going to be working with nanoparticles of 100 nanometers in size. So this little sugar ant is placed on a computer chip. This is a silicon wafer. And here is now the ant, much higher in magnification. And once again, we're going to call attention to how big is 100 nanometers. Well, right next to this ant is this little green box that's sitting on top of this silicon wafer. And my next slide is going to magnify that green box. And here it is down here. You see 2 squares that are over on the left side, but now at this higher magnification we can focus on features that are much smaller than the ant and the box, and these are little lines here. And this particular small line I've squared in the box, and the next slide is going to be an even higher level of magnification, and here it is. Well, this line here on this picture is this feature here. Well, that's not 100 nanometers. It's actually this little line that's right next to this big line. That's 100 nanometers. So I think you can appreciate as we scale down how small the features are that we're going to be dealing with. This is a 100 nanometer wide line, starting with multiple magnifications to get down to this. So nano is quite small. We also often have to use special tools and techniques to work with it, and we'll discuss some of those shortly. Okay? So now we get back to this concept of nanomedicine. It's an enormously important area, currently under very active research. And nanomedicine impacts both diagnostics, that is the detection of disease states, and also therapeutics, that is, as we'll see, nano-sized particles, okay, hold great promise for making significant advances in medicine. So I first want to just introduce or reintroduce to some of you the concept of a magic bullet. And this is where the promise of nanomedicine, okay, reaches, I think, the most significant levels of intensity. So the magic bullet was a scientific concept developed by a German Nobel laureate, Paul Ehrlich, way back in 1900. You have to appreciate that in 1900, this was before the very fundamental concepts of contemporary biology were understood. But he proposed a concept of a perfect drug, a magic bullet, that could cure disease with no danger of side effects. His observations came from, he worked with cells and looked at cells under a microscope, and he observed that certain organic dyes, when added to these cell cultures, certain dyes would localize in certain specific regions of those cells. And he inferred from that, with that simple concept, that, my goodness, wouldn't it be neat if you could design a drug that when added to cells or living organisms would locate at the site of the disease, okay, and perhaps cure it. So that's how this concept of a magic bullet began many, many years ago. A more modern interpretation is precision medicine, which is really an extension of Ehrlich's original ideas about designing a potent but selective chemical to take out a cancer cell. And so this little cartoon here shows this is a nanoparticle, 150 nanometers in size. Now, contained in this nanoparticle, this is an idealized representation of a magic bullet. Contained in this nanoparticle are drugs that you would like to deliver to a particular site, let's say a tumor in a body. Now, coating this cell are several features. These are the important features here. These are the targeting moieties. These molecules that we're going to attach to the nanoparticle provide the particle with a direction. It will locate and bind to some specific biological molecule. That's the targeting mechanism. There are also other things that are attached to this nanoparticle, and these are things that will help it slip through, okay, an organism, such as the bloodstream, to allow it to be delivered to its target, where it will bind by these groups, and hopefully, if all goes well, deliver the drug to the target area. So this is the concept of a magic bullet, and as we'll see, it's a wonderful concept. But there's been a somewhat slow progress in realizing this dream. And there are several important reasons which we'll discuss in a little bit. Okay? So targeted delivery levels tumor, the concept is to fabricate a nanoparticle that's decorated with targeting molecules that will take it to for example, to a tumor. And inside that nanoparticle, once it binds to the tumor, will release its payload of drugs, okay? And this would be an idealized concept of a magic bullet, targeted delivery to specifically a cancer tumor, okay? A few examples of how this can be realized, and this would be a study of in vivo molecular and cellular imaging with quantum dots. Quantum dots are small inorganic nanoparticles, typically 5 to 10 nanometers in size. The quantum dots are coated with organic molecules. Again, part of this is to ensure that these are compatible with the environment. And as I indicated on the previous slide, this quantum dot, which is fluorescence, that is, if you shine a light on it, it will glow a color. This quantum dot, which is protected with organic molecules, have molecules here which are targeting molecules. These are the molecules, when attached to the quantum dot, will help the nanoparticle seek out, for example, a cancer cell. There are also some other things that are attached, once again, to compatibilize this assembly so that it can sneak through, for example, without interference of other biological molecules and bind to the cancer cell. And again, this is one where the quantum dot is to image, in this particular case, a tumor cell. And so here's an example of in vivo molecular and cellular imaging with quantum dots. This is simply a higher resolution experiment than what was observed by Paul Ehrlich in his initial concept in 1900 of organic dyes localizing on certain areas of cells. So the quantum dots that have been decorated with targeting molecules now are injected into the tail vein of a mouse, and this mouse has certain selected tumors that have been implanted in it. And this image is shown just to show you that those nanoparticles can seek out okay, and bind to tumor cells in this particular case. And because the quantum dots respond once illuminated or irradiated with light, give off colored photons. And that allows us to detect cancer cells in this particular case. Okay? So the question is, if we would like to take a nanoparticle and deliver it, target to a particular for example, cell, or let's say a cancer cell or a tumor, how do we target specific organs or cells in the body? And that takes us to this concept of antibodies, okay? Antibodies are the current gold standard for targeting. And so we're going to spend a few moments talking about antibodies, how they work and how they're produced, just to provide you with a background to our research project, which, as you'll see in a while, is aimed at producing nanoparticles in the chemistry laboratory that function as an antibody. Okay?
Okay.
So here's the question: what are antibodies? Well, antibodies are large protein molecules. They're approximately 8 nanometers. Okay? They're produced by living organisms. You and I have antibodies that are circulating in our bloodstream. They're part of our immune system, and their function is to recognize and bind to other biological macromolecules. In our bodies, the antibody's role is to identify things that don't belong there, non-self. And if they recognize something that doesn't belong there, let's say a virus particle, they can bind to it, and that binding triggers a series of other events that, if all goes well, hopefully will result in the elimination of that foreign substance in our blood. Now, molecular biologists have taken advantage of antibodies. Now they're widely used in basic research, in industrial processes, in clinical applications, for isolation of individual proteins, for analytical or diagnostic purposes, and for research or therapeutic intervention. That's because molecular biologists have learned how to make these complex molecules, okay? Independent of, for example, humans or mammals, which contain antibodies. Okay, and I'll talk a little bit about how antibodies are produced, but just keep in mind that these are very complicated, large protein molecules. Okay, so again, I'll emphasize the importance of antibodies in contemporary science, in particular biomedical science. Antibodies are used as therapeutics, as drugs. They're used in research, all aspects of biotechnology. They're used for vaccines, for purification of other biological macromolecules such as proteins, and of course they're used as diagnostics. The role of an antibody is to recognize something. And what has been an important advance is that we can now engineer what a particular antibody recognizes. So here is a high-resolution X-ray crystallographic study structure of an antibody. This is the common antibody that you and I have circulating in our blood. It's an immunoglobulin G. Okay, I have a slide where this is actually rotating so that you can see it in 3 dimensions. But this complicated large protein, at this end and this end, they are the same. This is the business end of the antibody. Here is where the antibody, quote, recognizes foreign substances. Okay? So if we switch to the next slide, as I mentioned in the beginning, I'm a chemist. And so my comfort zone is thinking about things at the molecular level, okay? And so I like to understand, how does this antibody actually recognize and bind other things? Well, as I indicated, antibodies are proteins. They're composed of amino acids. And at these ends of the antibody molecule are amino acids that are presenting their side chains. It turns out that these are variable. There's about 110 to 130 amino acids at the ends here that can be varied. And depending upon what those combinations are, this eventually results in an antibody that can recognize something rather specific. These ends are referred to as the hypervariable region of an antibody. And I'll look a little bit more closely. Okay, so this big complicated protein molecule is capable of recognizing something. How does that actually happen? Okay, how do antibodies capture proteins and peptides and other things that they're capable of binding to? Well, once again, the interaction occurs between the side chains of amino acids that are protruding from the surface of this antibody and their target. So we look a little bit more carefully. Amino acids, okay, this is a protein molecule. The amino acids are just combinations of these. The side chains of the amino acids, which can be nucleophilic, they can be relatively greasy and hydrophobic, as shown here. They can contain aromatic residues. They contain charge, in this case, negative charge, and in this case, positive charge, shown here. They can contain hydrogen bonding donor and acceptor groups. So it's from the side chains of these amino acids in various combinations that the antibody presents to the outside world. And the interaction between the surface of an antibody and, let's say, its substrate or analyte is involved with collectively weak interactions. Those interactions can be hydrogen bonding, they can be electrostatic, they can be hydrophobic, polar interactions. These are not covalent interactions, but individually weak interactions which collectively over a fair surface area can result ultimately in a strong interaction or binding. There's also a complementary shape associated with an antibody and its antigen. Okay, and so I've just taken a few structures from the X-ray crystal literature. This is a fragment of an antibody, and this is its substrate. In this particular case, it's the protein lysozyme. So it's this interaction here, these weak interactions, hydrogen bonding, hydrophobic, electrostatic, when everything aligns properly, gives rise to a tight antibody-antigen complex. We point out that there are complementary surfaces between the antibody and the antigen, shown here. And once again, because I'm a chemist, this is again another X-ray crystal structure of an antibody. This is the surface or the binding region. And I've shown now in stick figures the individual amino acids that are presented to the outside world. And when it forms a complex with its antigen, its mate, these are the specific interactions, okay, complementary interactions that take place that result in binding. Here I've highlighted with this yellow ellipsoid the
Hello? 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 The sound disappear. Hello? Hello? Yes? Yes? Yes, I have your image. And the sound is it was some time ago, 2 minutes ago, very clear. We have the part program with the sound very clear. And now the sound author, it's more or less. Yes, you can continue. Yes. How are antibodies produced? Okay, all of these molecules is in nano size? Okay. Yeah. Yes. Please, doctor, you are finishing or we have more? Okay. No, it's if you prefer, we can do another interview to put more. Yes, if you want to continue, it's okay. Yeah. Or we have more? Okay, you can continue. Yes. Yes. First, thank you so much for this class about this area of the nanomedicine. I think it's was so good question, so good class with many details that I did not never have one interview about this. Apply at this kind of subject. So I would like to invite you again to start in the last 10 slides, and because I didn't see the collaboration with Brazil in your research. Yes, I saw it. But here in São Paulo, we have a very old institute of research with snakes. The name is Instituto Butantan. That is the institute that produced the vaccine for us. But I don't know yet, until now, if the institute is working with this kind of research as you show for us. I think the people is not working with nanotechnology and this kind of research. But I can try to discover if there is some people there working with this line of research. Because it's, here in Brazil is very important, this Butantan Institute, because it is the only institute that produces vaccine for many kinds of snakes. And I would like to have another interview with you to discuss more in detail this possibility of this kind of project. And if I understand, it's possible to try to discover one possibility for many kinds of or many species of snakes. So I think this will be possible in the future, near future, I don't know. But I think this is one very important contribution for the people, because the people that have snake bites in general is in rural areas here in Brazil. In the city is not so common. In the city we have another problems, not with the snakes. We have another problem with the other kind of possibility to be bitten by another kinds of species. But the snake is from people that is living in the rural areas, and in this space there is no antidote. So it's very common that people that is bite for snake in the rural area have problem for the life if you don't that. So I think it is very good your project, and the team that is now the young team of research that can contribute with this. I have another question, but I prefer to put for you in the next interview. I don't have knowledge to put in deep question for you, but you put in the part of your presentation about the molecules that are interested by the companies. No, the companies are interested in many types of molecules, and I don't know if in this kind of solution the companies are interested, because it's for the companies in general is small people, a small group that have problem with snakes. I think this companies is more interested in cancer. Huh? So this kind of solution, I think it will be difficult. You have a solution from companies. Okay, Professor, thank you so much for this class, and I will send you new email to propose a new interview. Okay? Thank you so much. Bye-bye.